spend a lot of our time thinking about new peak detection, its reproducibility, what parameters are most effective to use to compare to a reference standard. I think that one of the challenges is there's no sort of streamlined approach to sort of form new peak detection. The other thing, right, is from my perspective, what it depends where in the product lifecycle you are. What does a new peak mean, right? And and that's part of the reason why I think there's some debate too on, as you move down the product lifecycle towards commercialization. If you get to the quality manufacturing space, if you have a new peak and you don't have the proper instrumentation to characterize that peak, and you have to go back to the beginning in sort of a research mode, that's something that people are debating, right? Simpler instrumentation in that quality space is great, but if you get a new peak, what do you do? And the question that I spend a lot of time training regulators around the world, and people always in industry, but people always ask me, like, if you saw a new peak, what's the threshold that we should be concerned with? And I don't have an answer to that. I don't know that anybody really does, but at this point, but it's except to say that it would be product dependent. But really thinking about where it would be really wonderful if the industry, not just companies, but the totality of the industry, government, academics, and industry companies that sort of drive in the same direction.
NPD is really required for establishing the purity assays. And this is really a good tool to flag out the outliers, especially when you're comparing a number of samples like a large base, because that's the need. Like what Jared said, there are challenges in implementing this, and what threshold should be defined, like, what is the real cutoff value, so that you are able to identify that the new peak which is identified is actually not an artifact from the electrospionization or various other modes, or it's actually a new peak. So that's the debate which is happening on what should be the defined thresholds, how to really make sure that the peak detected is not coming from an artifact. And that's where, when you're defining the libraries, I think that will come to a handy. If you have the flexibility in the to define that, any new peaks coming up, you identify that, characterize that, and put it in your library so that next time this can be taken care of. So again, a lot of optimization is still needed, which will help in identifying those outliers when you're working on.
One of the biggest things I think is we just need more data. I mean, we have a lot of purity methods that are already accepted by the agency, by industry. And so if we're going to kind of change how we're thinking about purity and try to use this new approach, we're really going to have the burden to show that it's as good or better than what we've been doing previously. So kind of figuring out the failure points or those peaks that we are detecting by our other methods, and showing that correlation to NAM, and showing that concentration correlation, that percent impurity. We really have to rely on the data we have so far to kind of figure out what targets that would be, and I think it's going to have to be a continuous open, especially for regulators to try to figure out it's acceptable here. And with NAM, we're talking about mass spectrometry, and as I'm sure all of us know, we do see fluctuations in the intensity of your analytes, from day to day, from an analysis to analysis. So it's not as, I don't want to say it's not as repeatable, because it is nominally, like within a sample for sure, but when you're talking about different days. So setting those thresholds, it's really challenging, because you might get an 80,000 response one day, and that's going to be 100,000 another day, and we all know that's fine. And then how we expect these systems to operate, when you're trying to use that to make this like standardized threshold, it becomes really tricky. And then, just going into as well, I know some people talk about new peak detection, but they don't actually mean new peak. They just mean increase of a peak they've already seen, or decrease of a peak, and that's not the same thing.
We definitely need more data. We need it to be more reproducible, but also there's got to be a lot of education to regulators on, what the data is, what it means, and what any variation might mean.
Yeah, I think, and to add to that point, just defining the threshold won't help. So there can be like multiple other attributes which can be monitored along with the threshold to be defined. And then the question is whether to define absolute intensity or the relative intensity. So you're looking at the fold change. So like what Rachel said, that the type of variation you see on a day-to-day basis, and it's not something like you are monitoring on. This needs to be done for a longer time, for maybe months or years. So then defining those relative values would help. And along with that, you can have other parameters, attributes like maybe isotope distribution, maybe the molecular weight. So like that, you have to have like multiple parameters to define in those new peaks identified to make sure whether it's a false positive or a false negative, to make sure that it's a true identification.